When you look at the genome, it's 3 billion letters long. And if we think about the genes that most biologists study, these genes only make up about 2% of that 3 billion letters. And so the big question in genomics is, what is all the rest of this genome doing? It turns out that the vast majority of that empty space actually derives from these ancient genetic parasites known as transposons. And these got in there throughout our evolution and embedded themselves in our genome. Most biologists perhaps rightfully assume that these sequences are not necessarily functional for human biology. In a healthy cell, really most of these sequences are silenced or inert. But now we know that in many diseases, including cancer and aging, these genome defenses just break down. These transposon sequences that are normally silent in a healthy individual all of a sudden become reactivated or awakened in these disease settings. We actually found one example where there was an ancient retrovirus that was required for turning on a DNA repair gene in colorectal cancer. And that actually helps these cancer cells survive against many kinds of chemotherapies and radiation therapies. We took colorectal cancer cells and we just removed that one ancient retroviral sequence. And what that did is it turned down the expression of that DNA repair gene and also made those cells more sensitive to radiation therapy. The Packard Fellowship was really a transformative opportunity that we received early on in the lab's history. And I think so much of our success then and now can really be attributed to the generosity of the fellowship, but also just the freedom that it gave us to pursue the questions we were interested in. Moving into the cancer field was really something that I hadn't planned for. When I started the lab, my training was really in genomics and really specifically evolutionary genomics. By following the science, we realized that the genes that we had found also had this tremendous therapeutic potential. Probably more than half my lab works on cancer in some fashion, and this was something totally I would not have predicted when I started the lab, but I'm really proud of and really excited about.